We established in the last lecture that the vast majority of mutations that occur are simple substitutions or insertion deletions, right? Small mutations, we call those point mutations, where just one base pair changes. We also established this pattern of mutations where the vast majority of them are effectively neutral, maybe 95% or more. Again, those are my numbers, but I think it's a pretty good estimate, albeit not very precise. It is quite accurate. And of the remaining 5% or less than the mutations, the vast majority of those remaining mutations are negative. It's something that impacts the organism badly. They don't do as well as they did before, or as if they didn't carry that mutation. And then finally, there's this tiny fraction, probably less, maybe even way less than 0.1% of mutations, which are beneficial, which provide an adaptive advantage for those organisms. Okay, but that's where we're starting from. Now, let's look at why the vast majority. So we're going to spend a little bit of time here looking at why 95% or more of all mutations are neutral. Okay, why don't they impact the phenotype of an organism? Okay, and this stems out of a revolution in the way that we are able to look at data. You know, the fact that we have vast amounts of genetic DNA data available, we can compare it between many individuals in a population, and we can compare it between different species even. But even before we had those vast amounts of data, theory and the very early hints of some of this molecular data began to lead people down this pathway to understanding that much of what occurs, in fact, the majority of what occurs in diversity in populations is not good or bad, it's effectively neutral. Okay? The number one reason for that is non-coding DNA. This is especially true for eukaryotic organisms, but there is some non-coding DNA in prokaryotic organisms also. But in eukaryotic organisms, especially mammals, birds, well, all multicellular eukaryotic organisms, there's a huge percentage of the genome that is non-coding. That means it doesn't code for protein products. Okay? And in humans, this is about, here's a human pie chart of our entire genome. About 98.5% of our genome is non-protein coding. So there's that little tiny slice, 1.5%, that codes for genes. And this is by and large what causes our phenotype. Now realize it's not everything that causes our phenotype. Within this vast space of non-coding DNA, there are some important components. Some of them are structural and just need to be there for proper replication and segregation and cellular division. Right? So those are important, but they don't code directly for a protein. And other parts are regulatory. And then there are other things like microRNAs and a lot of things, that some of which we're just beginning to fully understand. So when I say non-coding DNA, don't think automatically, oh, it's not important at all. There are parts of it that are important, but those important parts are a very small proportion overall of the non-coding DNA. So if I randomly mutate the non-coding DNA, the odds of me doing something that's going to impact the phenotype, either positively or negative, are very low. Here's an analogy. Imagine you dropped a large boulder on the state of Texas. What are the chances that you injured somebody if it just randomly dropped somewhere in Texas? And your odds are pretty darn low, right? Depending on the time of day, but you know, well, I guess probably time of day wouldn't matter if you're doing it over the entire state of Texas. Your odds of hurting someone would be about the same, and they'd be pretty low. Now, what if instead we said, let's drop a boulder on Manhattan? Now, suddenly, we still might miss everybody and do some damage but not hurt anyone, but suddenly our chances of injuring someone are way, way, way higher if we drop it on Manhattan. And that's a good analogy for mutations in non-coding DNA, like dropping a boulder on the state of Texas, versus mutations in the protein coding regions, like dropping a boulder in New York. We have a much higher chance, although not 100%, of having a phenotypical impact if we mutate a protein coding area. And in these non-coding areas, the chance is not zero, but it's very low. So most mutations in non-coding DNA are there. You can measure them, you can use them for other things, but they don't impact the fitness either positively or negatively of the organism that carries that mutation. Reason number 2, even in the protein coding regions, many of the mutations are silent, which means a silent mutation is any mutation that doesn't impact the phenotype. So most of the mutations in the non-coding DNA are silent. Now, there's a special case of silent mutations in the coding regions called synonymous mutations. So think of synonymous mutations as a subcategory of a silent mutation. And this has to do with the genetic code, which we'll review, but hopefully you're already familiar with. We're going to review it in a couple of lectures. And basically, because of the genetic code, most mutations, about 85% or so, at the third position in the reading frame are effectively neutral. So GAU codes for aspartic acid. I think that's what that abbreviation is. GAC codes for the same amino acid. Glycine is coded by either GGA or GGU. So at the third position, it almost never, vast majority of the time, if you mutate it there, it doesn't matter. This species carries the new mutation, but they still put an aspartic acid at that one place in the protein, or threonine or an isoleucine or whatever it is. They put them in there and boom, it's exactly the same. And so notice that although there is diversity at the DNA level in this protein coding region, there's no diversity in the amino acid sequence. And that's a very common pattern that we'll see in many, many genes across different organisms. And it has to do with the degeneracy of the genetic code. Now, don't be confused by that word degenerate or the degeneracy of the DNA code. People immediately put that, say, oh, that's a bad negative thing, that the DNA code is degenerate. It's degenerating. That's not true. It's a different usage here. It may be redundancy of the genetic code is a little bit better, or the fact that there are multiple synonymous codons for the same amino acid. Okay, so think of it that way, and if we mutate the third one very frequently, we have no impact. Now, occasionally the first one can mutate and still have no impact, but that's less common, much less common. And the second one, every time you change the second position, you always change the amino acid. Okay, so that third one tends to be where these mutations accumulate because natural selection removes most of the mutations that happen at the first and the second place. So let's expand on that just a little bit and think about why. I just don't want you to take that for granted. Let's think about it. So if we randomly mutate the third position, we already know that our best chance of what's going to happen is that it will be synonymous, which is a silent mutation, and therefore natural selection plays no role at all. Doesn't remove it, doesn't keep it. The only thing we have going is genetic drift. So genetic drift would keep some of them, remove some of them, and really wouldn't have a strong force one way or the other, but would randomly fluctuate it. And so over time, we would have diversity building up at the third position because they're neutral and some of them are allowed to stay. Another way to think of it is mutations at the third position have very low exposure to the effects of natural selection. Now, what happens at the second and first? We'll treat them roughly the same, although the second's a little different because some can be synonymous, but a small number. But for simplicity's sake, a mutation at the first or the second position is almost always, and at the second position, always going to change the amino acid. So now we have a different question. What happens when we swap out one amino acid for the other? And if we're playing the odds, it's probably going to have some impact on phenotype. And again, playing the odds, if 5% of all of the mutations that occur and change the phenotype are negative, and less than 0.1% of all the mutations that change the phenotype are positive, almost everyone, the vast majority of mutations that are at the second and the first position are going to have a negative impact on the phenotype of the organism. The fitness of that organism is going to drop. And so natural selection will work to remove variation at the first and second positions most of the time. And there are special cases where that's not true, and we look for those, and there are exceptions to the rule, and they're exciting because of that. We'll talk about that in coming lecture. Reason number 3 why most mutations are neutral. Sometimes a mutation does change the amino acid of the sequence of a protein, but it doesn't impact the function of the protein. And in fact, there are parts of a protein that are much more conserved, meaning that when mutations occur there, natural selection removes them. And other parts where natural selection doesn't seem to care so much, even if amino acids change, they can be kept. And so areas of proteins that are very, very highly conserved between distantly related species show that, well, let's look at this. There are 2 reasons why that could be. Number 1, it could be that there are very, very low mutation rates, that we just never get mutations in that area, and so they've stayed the same. Now, from what we understand about the nature of mutation, that's very, very unlikely, because mutations are random. So mutations are happening equally across all parts of the protein. So then the question becomes, If mutations are happening equally, why do we see less diversity in some parts of a protein? Those are highly conserved, and much more diversity in other parts of a protein. And that's because of what we've been talking about. When a mutation changes the amino acid in these conserved regions, natural selection works to remove it, and we never see that diversity because it's removed from the population. However, when mutations occur in not really very important parts, maybe we change a leucine for an isoleucine, maybe we change a glycine for a phenylalanine, whatever it is, and it doesn't impact the phenotype of that protein, doesn't impact how it works. And so we build up diversity in some areas and very little diversity in others. Okay, so these blue areas would be areas where neutral mutations can accumulate despite changes in the amino acids. And finally, our fourth reason why most mutations are synonymous is that even if we change the phenotype, even if that phenotype results in a shift in fitness, which it doesn't, maybe I don't know if eye color, there's some evidence in some populations that eye color in humans might be slightly under natural selection, but probably not enough to be significant, enough to play a critical role so that it is outweighed by random mutation, or it outweighs random mutation or genetic drift. So maybe we have changes in phenotype that impact the fitness, but it's so insignificant, it's a measurable amount, but so small that it doesn't really have an impact into our overall calculations about the diversity of organisms. Okay, so review and make sure you know those 4 reasons why the vast majority of mutations are effectively neutral.